Hello.
Hi, Bradley.
Hi, how are you?
Hi.
How are you, Susan?
I have seen the corrections in the doc.
Yeah, I haven't seen the doc recently. I made
Yeah,
some
yeah.
comments last week, but
Okay. I mean, last week I haven't touched the doc. Actually, I was stuck with one technical stuff of loss function. I was updating all the week that code only. Finally, I somehow got it work. This week update would be that. Could I share my screen and start?
yeah, go ahead.
Last week,
Here we go.
completely I was working on the loss function. I wrote this all code, which are looking as green here right now. This all is a part of loss function. I have implemented different loss functions, and I'm comparing between them which loss function will give the best results. Right now, I have the dice loss, dice BCE loss, and to implement this, I need to read a paper actually. Uh, yeah, I have read this paper and I have implemented it. Unified focal loss. This is about loss function mainly used for segmentation. This paper was completely dependent on that only. Uh, it mainly focused on a particular loss function called unified focal loss. It is an intersection of dice loss and cross entropy loss. I have read the whole paper and I understood and I have implemented their methods in the loss functions. Uh, right now, whatever I could have was the dice loss was a common function everyone will use. I added the dice PCE loss, IOU loss, IOU PCE loss. I mean, different things, symmetrical focal loss. But unified focal loss is a mix of two loss functions. One is asymmetrical. One was dice loss and one was PCE loss. This complete week took around time around this only, reading the paper, understanding it, implementing, commenting every code, how it is working and extra. Mike told me to write code in this way, like parameters, what will it return, etc. So
Yeah.
it took a couple of days to implement all this stuff, learn about it, and these loss functions are not available over internet. Actually, generally, loss functions will be available in the PyTorch library or something like that. But this unified focal loss was completely different. I couldn't find it online, so I wrote my own code and I made it work. It is working right now, actually. This code whole part. I will try to. This will be my this week update would be this only. And yeah, I haven't seen. Actually, you told Bradley that my GSOC period will be extended on Monday. Haven't seen check today, but yeah, hopefully it get extended.
Yeah.
And this is my update. And this week I will be writing the doc about the code what you have mentioned and all stuff, and implement all these things. There are a lot of things I need to implement it. Uh, yeah, that was my pretty update all about. And these loss functions are mainly used in the DevoNet, not in the SAM. So these loss functions will be compared which loss function will give us better output. So I'll compare which one will give better output and I will implement them. I'm adding the project structure in this way. All parameters will be taken from one file and those will be implemented code directly. No one needs to run some terminal commands. I am using argparse library. Uh, yeah, this is a Python library which is used for uh, command line arguments. We can keep our all whatever we need in a file and those will be taken as the command line arguments. We can change, we need to, if someone wants to use DevoNet, they just need to change the parameters here and just run a one line of code. It will train it completely by itself. So that was my implementation. And yeah, pretty this week I have worked on the loss functions only. And in coming week I will implement on the I will see the suggestions you have made in the doc. I'll try to implement. I will try to edit them and I will change them and I will go on with the coding thing which I need to do.
Okay. That sounds good. Could you bring up the
Okay.
doc if you wouldn't mind? Yeah, thanks for the update.
I will change all these which you have mentioned right now, because there are a lot of things I need to change. I haven't seen the doc actually after you mentioned it, because due to this paper, I was reading this paper and trying to understand, so it took some time. There are a lot of technical terms I need to understand before implementing this all stuff. So, yeah.
All right.
I will read these comments and I will try to implement it.
All right. Yeah. So actually, I want to make a couple points. So the methods are kind of like where you put a lot of the, you might kind of like walk through like how the program works, have some of the features. So some of the things you're showing in the meeting, you might highlight those in the methods. You might have like a heading for, say, like the loss function, or for some other thing that you're working on or that you've worked on. Describe it a little bit. Describe, like, cite some references like you showed, and just go through kind of, go through the sort of how it works, sort of at a high level, and then get down to the specific things that you might want to highlight. So things that are unique about this platform,
Okay. I will mention the method I have used, and the results I will compare with other models. Actually, we can take for example, we can take unit. I will compare our model results and unit model results. I'll compare our model and DevoNet. Yeah, that's the thing only, right? Results will be.
Yes.
Okay.
That would be good.
Future directions I will see. I don't have anything in my mind right now about future directions, but I will see what we can implement in the future.
Yeah, I would wait. That would be kind of one of the final things, though, because once you get through the paper, you'll see kind of how that shapes out to be. Like if you describe what the platform is, the different things that you worked on, describe the results, and then think, what's kind of the next step here? What is it that we could do next? And we've talked about some of those things, but some of those things are not like obvious until you actually go through the results. Because sometimes there will be things that you'll notice in the results that are like, oh yeah, we need to work on that. Then usually when people do, like future directions or things like that, they'll usually think at two levels. They'll think at sort of like the immediate level, where like if you look through the results and you notice there's something that isn't really resolved, you might say, well, we want to do this to improve the loss function, or we want to do this to improve the performance of segmentation. And then, that those sorts of things are the first kind of immediate level future direction. Then the next level is like, how do you want to integrate this into some other system? So, it might be like encouraging people to use it, or improving the UI, or integrating with other tools, that sort of thing. So usually that's how people write it out, and it's just we'll just have to think about that a little bit. But I wouldn't worry about that until the end.
Okay. Try to implement that. Okay. First thing I have in the mind is we need to integrate with lineage population. When we give a direct image, that would be our immediate effect, would be next project of extension of this. We are trying to segment the cells, right? According to the cell positions, we'll try to extract the lineage population. I have read these papers actually. In that nomenclature of A cell, B cell, or something like names given are based on the position of the cell. If they are located at anterior exterior position, it is called as AE. Something like that. I am trying to understand it. Yeah, our next project would be that only.
Okay. Yeah, that's good. And then you might also, include some examples of code, of course, in tables. Like you'll put a table where you'll have a little bit of code in there. Usually it's just to show an example. It's not like to show the entire code base. But and then, of course, you'll want to put down the different models that we have, the lineage population, and some of these other things. Kind of like those would be like at the first part of the results, just kind of like, these are the components. This is how they work. This is where you can find them. So we have the Hugging Face, and we have the GitHub repositories. So, yeah.
Okay, fine. I will implement them. Thanks.
All right.
Can
no Yeah, it looks great. Thank you for the update. Yeah, I talked to Himanshu this morning. He's feeling under the weather, so he's not going to join us. He's got, I don't know, like some sort of health issue, so we'll get an update from him soon.
Is Lukas coming on?
What was that?
Is Lukas going to come on soon?
I'm not sure. I haven't heard from him today. I was
He said he
talking
was
to
going
him.
support today.
Oh, yeah. I was talking to him this last week, so Lukas and I have been working quite extensively on some of the bioinformatics stuff that we're working on for the paper.
We need an explanation for the uninitiated.
Yeah. So, Susan, would you like to give any updates, or
Give a what?
give any
Update?
updates on some of the things you've been doing?
Oh, I've been having issues with my models again. They're stable, and then all of a sudden you add more force, and they go like
Oh,
this.
yeah.
So when there's a big jump in the displacement that they experience, and after that, sometimes they're just linear.
Sounds like you shouldn't live in a tensegrity house.
Well, I'm not too sure how to graph that.
Right.
Because the J-curve you're looking for is in the first little bit before it collapses. Well, it doesn't really collapse. It just changes. Well, all of a sudden.
Right.
Poof. Yeah, and I've got the microscopic ones, and they don't behave like the larger versions. It's like, oh no. And I don't know. It's supposed to be the same. Like there's no nanotechnology attached to the trusses, but still,
Right.
I get anyway.
Maybe they're round off errors.
I'm not sure what's going on. I have it in micrometers, and I have it in millimeters, and the millimeters don't seem to be the same as the
Try
micrometers.
parsecs.
Pardon?
Try parsecs.
Parsecs? Oh yeah, sure. I wonder what that would look like. I could try it. The program would probably go bonkers because I tried an infinite elasticity by making it 10 to the 18 Pascal of elasticity, and I was get error. It would calculate it for me, and then it would go error.
Sounds like round off errors.
Pardon?
Sounds like round off errors.
Oh, maybe. I'm not sure, but it was giving me the same graph as it was before, and it wasn't supposed to.
Okay, Susan, we need you to think through whether or not you are making a contribution to this paper, so whether you should be an author.
This is the paper on differentiation codes in C. elegans.
Yeah. I don't know. What you tell me what I can do for you in the way of modeling on Comsol, and I'll tell you whether I can do it.
Yeah.
Well, unless you can model a nucleus, I don't know.
Well, I might be able to model the nucleus.
Okay, we got Lukas there.
Yeah.
Hello, Lukas. Oh, hello.
Hello.
Anyway,
Yeah.
so yeah, I'm still modeling, and I'm supposed to be done by now, and things are just annoying.
Yeah. Well, good luck with that. Yeah, it's always a struggle to try to get the right parameters and figure out with these programs.
I pitched a tent on a boat.
Yeah. Yeah. So thanks for the update, Susan. And good luck with your camping trip. And
Yeah.
A tent on a boat is it acts like a sail.
A what?
Tent on a boat acts like a sail.
Oh.
I have that from experience.
Oh. As long as I don't get seasick and have mosquitoes bite me at night, I might survive the trip. Yeah. Anyway,
Depends on whether you're on a river or a lake.
we're on a lake. It's called Lake of the Woods.
Oh, okay. Should be okay. Enjoy the fish.
Yeah.
I'll watch them, take pictures. Can't eat fish.
Yeah. So yeah, we've been working on this paper, the bioinformatics part. So Lukas has been working on the the protein blasting, and then I've been working on some other things, and we've been talking about how to make comparisons and matches. So Lukas, did you want to talk a little bit about what you've been doing?
Oh yeah. So what we did after I talked to Bradley, we came up with we came up with a couple of things like so what we did was we set a setting like algorithm like variables, and so there's a couple of like variables in the protein blast. One of them is max target sequence, and after the discussion, me and Bradley discussed it and said that the max target sequence for all of the blast experiments, like blast searches, would be 250, and the E threshold would be 0.05. And what we've been doing is, what I've been doing is just blasting all of those 23 proteins against the proteome of C. elegans, the rat, and Drosophila, like the fruit fly, and the other one was mouse, I think. Yeah, so I've been finishing the blast for the fruit fly, and now I'm continuing with that. I couldn't finish it this weekend, but I've been working on finishing. Like, I think I sent Richard as well. Like, I sent you a file on it's called bioinformatics details, and I want to finish it by the end of the week, and we could discuss all of the four results. But as of now, I am actually facing a problem because I couldn't find the.
Go ahead.
Yeah, I've been facing like a small challenge because for the beta catenin protein, I couldn't find any protein sequence for the Drosophila one for the fruit fly.
Yeah.
But yeah, I've been discussing some interesting proteins with Bradley as well. So we're gonna. I've taken note of those ones as well. So the interesting proteins. So what I do is I run all of the blast search, and I check all of the protein names, and if I see something interesting, I want to take note of it as well, so we could discuss it. So for this meeting, I think we can discuss this protein. It was this one. So when I was running the blast search for just a second. When I was running the blast search for CK1 protein for Drosophila, and when I compared the sequence against the proteome of fruit fly, I saw an interesting match, which is this protein, Balchan protein. I don't exactly know it, but what I found is it has an involvement in chromosomes cytoskeleton organization, and that might be interesting to just discuss for today. But yeah, I think Bradley also sent me. So by the end of so when I finish the bioinformatics details, I think we're gonna so I'm gonna also copy what Bradley out here. So I think by the end of this blast search for all four, we're gonna I think we're gonna pretty much see the same thing that Bradley said. So like mosaic development B for C. elegans and A for Drosophila, and the regulative development for mouse. So these blast search I think would also help us with they would support the Bradley's like what would you call it? Bradley's statement here. If we have mouse and those, I sent it there as well. So yeah, and we could discuss these.
All right. So put something in the chat. A couple of things in the chat. So the first thing was, of course, the beta-catenin. So a lot of times in different species, it's not always the same thing. They don't call it the same protein, or there's a homolog, which means it's a sort of a divergent form of that thing, but it has a different name. That's this is nomenclature a lot of times, like in C. elegans and Drosophila, the genes have different names, but they're basically the same thing. So we can find out if you could make a list when you're doing this analysis. If you see something that doesn't match, send it along, or create a table is a better strategy. And you update the table, you update the numbers in it, and then if you have something you can't find, we can look at you can just search for beta-catenin Drosophila, and it might give you the alternate name for it, and then that would be the thing you'd try to search for. And then in the table you would have a note that would say this is also called this in Drosophila. So you can see the similarity in the sequence, but it's not going to be the same name. So that's how you kind of clean up some of those entries. When we do comparisons, sometimes we're not able to compare directly, so we have to make these indirect comparisons.
Bradley, funny story. Natalie was once reviewing a paper where the guy had a different name for some gene. And she pointed this out and said, well, if I don't give it a new name, I can't get a paper.
Oh, yeah. Yeah. I mean, a lot of times, it's just people are working on it. When in C. elegans, there's a community of people who have characterized the genes and everything. And then Drosophila has another community, and they don't necessarily talk to one another about, is this the same gene? Because remember, a lot of these draft genomes didn't exist when they were naming the genes. So they were just sequencing genes, one at a time, and they were trying to figure out what they were. And so this is where, there's no overarching nomenclature. So, but sometimes there is. I mean, sometimes people know what something is and they give it the name, but sometimes there isn't. So then there's Balchan protein involvement in chromosome cytoskeletal organization, which might give some clues. Yeah, please make that note of that. And then, try to maybe do a search for that. If you find interesting things, you can do a search and get the numbers for that.
Yeah,
and we can actually see.
Question.
Yeah.
What has anyone investigated differences between the asymmetric divisions for different cells in C. elegans?
I don't think so.
You don't think so? Okay,
No.
because you can't say all asymmetric divisions look the same because they result in different cells.
Right.
Okay, I'm just throwing that as a place to possibly look, because the asymmetric division is analogous to the cell state splitter, and so it could be the spindle apparatus is what is different between different cells, even though it looks the same.
Yeah.
So I don't know, can you get a list of spindle apparatus genes for C. elegans?
Yeah, we could do that. I'll look in the. I'll see if I can find something.
Okay, Lukas, you're going to drive us nuts. The pronunciation for the fruit fly is Drosophila.
Yeah, I just realized it. Sorry.
Yeah.
We'll translate every time.
It's like, yeah. Yeah, so Drosophila, yeah. So Drosophila is one of our comparison groups. Drosophila is actually a taxon. So it's like Canorhabditis. We have Canorhabditis elegans, and we have some other worms that are related. And actually found a good paper on that this week. I'll probably talk about it after. But yeah, so Drosophila, the one we're usually interested in is melanogaster, which is just it's the model organism. So it's like C. elegans. And then there are other Drosophila species, and I think those have been less characterized by like models at the model organism sort of level of specificity. But there have been a lot of, so you may find things that are Drosophila spa, or which is SP period, which means they don't know what the species is, or some other Drosophila species. If you can find Drosophila melanogaster, then that's the thing you go with. But if it's Drosophila spa, then that would be okay, especially in this context where we're comparing with C. elegans. So it's like obviously there's a difference there, quite a big taxonomic difference. So
yeah, for all of the proteins, I found it within the species of melanogaster, I think.
Yeah.
Okay, can I make a suggestion that
Yes.
you retain all searches and then rank them by the number of copies found
Okay, sure.
in a table?
Yeah, I'll do that.
That would be a great contribution because it shows what's possibly relevant and what may be irrelevant to the question of differentiation tree.
Yeah, and what Bradley also said is we could then, like, after at least the proteins, we could also compare, like, look at which ones are involved in the differentiation, which ones are not. So that might also help a bit with the problem.
Well, yeah, I think we can judge from the annotations, like which ones are sort of maybe more central and not. I mean, it's going to be subjective because the annotations are sort of based on observations, maybe, or like I don't know who's generating the annotations, and usually they do like they'll just cite like studies, and that's the annotation. So it's like if you notice the annotations are rather broad. Like it'll say phosphorylation, or it'll say cytoskeleton, and it doesn't get much more specific than that, and that's okay. I mean, that gives us clues to where what's going on, but I think I don't see anything that's really not involved. Obviously, that was the list that Dick had from the book, but they all look like they're probably going to be somehow involved in differentiation, just like there are different types of things. Like there's phosphorylation proteins, there's different cytoskeletal proteins, and so those actually could be compared as well. And I don't, we don't really know. Like there's a lot of genomic data, a lot of molecular data being generated, and a lot of it's been characterization up to this point. So when we say something about function with a lot of molecular data, unless we're doing like a specific experiment, it's hard to really kind of make definitive statements about what that looks like. I mean, there are very few studies that actually show like a broad study like that where they say, these proteins are involved in this process, these proteins are involved in this process, and let's make a comparison. I mean, it does occur, but again, it's like usually it's pretty focused on specific questions. So that's something we'll have to sort of work around too, but that'll be for later. So there is also a lot of structural stuff to be expected. Cell adhesion, microtubules, etc. at high counts. Yes, there should be a lot of structural stuff because when you do differentiation involves cell division, and that's going to happen. And you have to, in fact, you have to have a cell division a lot of times to have differentiation. I don't know if that's entire. I mean, well, you do have to have it to get the. You'll have to at least replicate DNA to have the things expressed that you need to have. So it's tied to differentiation, cell division, and some of the other, and things like cell adhesion. Cells are moving around, they're changing their shape. They're those are going to be important things.
If you want a picture of where some of the molecules are and their function, you can look at the paper there, because it's got the alpha and beta catenins and microtubules. I don't know if it has microtubules, but anyway, it's got actin filaments and where they're placed in the cell, sort of. It kind of shows you like the physical layout in this paper. It's an older paper, but that's okay.
Yeah.
Okay, Bradley, question.
Yeah.
Are there any genes that Mike Levin has associated with the bioelectricity phenomena?
Yeah, I think so. We could look that up. I don't know what they are right off the top of my head. Okay. But yeah, that's another thing.
Be involved. I mean, for example, Lionel Jaffe showed many years ago that there's a huge electric current that comes out of the neural plate of the chicken.
Yeah.
Okay. So there might be some relationship between electric currents, the genes that cause them, and the differentiation tree.
All right. Yeah. And so, yeah. So I also see some interesting counts for things related to phosphorylation, related to differentiation, induction, and human stem cells. Yeah, we went over some papers on this. So phosphorylation is important in a lot of things in the cell, especially for molecular aspects of the cell, like for function. So you have these phosphorylation cascades that help in function and some functional aspects. So I sent them a paper on human stem cells where they had this. They found a lot of phosphorylation related to differentiation, induction there. So this is like, I guess, a naturally differentiating cell that is going from a stem cell to a precursor cell. Not every differentiation related thing is a high count, however, and that makes sense in light of what I said on Monday. C. elegans exhibits mosaic controlled differentiation. Yeah, and this is why I wanted you to look up the mouse data, because you would have, so you'll have like basically 4 categories. So there's mouse, or there's C. elegans. So let me write on the board here. If I'm sharing my screen, I'm not sharing my screen. Let me share my screen. So you're going to have 4 categories that you're comparing. So you have C. elegans, which is mosaic development. You'll have Drosophila. You'll have, I'm running out of space, mouse, and you'll have yeast. So you'll have a mosaic development here. You'll have another example of mosaic development. So we can say if maybe some things that are involved in differentiation are, being produced and some are not, then that's just mosaic development, and that's site dependent. If we see a similar pattern in Drosophila, but maybe not the same proteins, then we can also say that's another example of mosaic development. In mouse, you have regulative development. And so that means that you should see like a more consistent pattern. But we don't know. And then yeast would be no development. So that's where you don't really have a developmental stage. So you may have those proteins, but you don't have the same pattern of expression of genes and then proteins. So that would be kind of the idea of all those.
Bradley, can I ask a question about the mosaic? Remember, I don't know if you know the old concept of commitment, okay, where if you transplant a cell within an embryo from one place to another, it does one of 2 things. It either becomes similar to the neighboring cells, or it develops independently up into a structure.
Yeah.
And this was presumed to be evidence that at some stage a cell became committed to its differentiation.
Right.
Have those kinds of experiments been done for mosaic organisms?
I don't think so. Yeah, I don't think they have. I know there's this one paper where they've dissociated the 2-cell stage and they formed like their own little cell culture of C. elegans embryo, and they just form like a weird pattern on each side, and I've never seen anyone take like a cell, put it in some other part of the organism, and then it takes. Of course, the flatworm stuff, you can take a single cell and it can generate the entire organism, but that's a different thing, I think.
Yeah.
Pluripotency.
Okay. Well, maybe put you back in the lab.
Yeah. Well. Yeah. So yeah, that's the logic there, and then so this is where Drosophila would come in handy, and perhaps even mouse. If we had mouse, Drosophila, C. elegans, and yeast for all candidate proteins, we would have no development yeast, mosaic development A, mosaic development B, which is Drosophila and C. elegans, and then regular development mouse. We could then evaluate the differentiation and non-differentiation related proteins to see if they fit that model. Yeah. And so we have the protein list, we have the different organisms, and that's what Lukas is working on now. And yeah, just try to get it done when you can. I think by the end of the week would be a good timeline, and that then we can look at the results in total. So
then it's time to start writing up because we'll only have 2 weeks as well.
Oh yeah, well 4, right? Because it's at the end of August.
August, yeah, August 30th.
Yeah. So yeah, that's good. And then I'm working on some additional analysis just with C. elegans. So I think last time I showed you the paper on the C. elegans browser, which is where you can look up genes by their, like, you can look up a gene, you can look at the locus, and you can get the sequence on the chromosome. So it's a very nice tool for looking up specific genes. What I'm doing is I'm going back and I'm trying to find the genes for different proteins. And then I'm looking up the CG motifs in that locus. So I don't know where the promoter is, and I don't know where the coding region is necessarily. But what I can do is count up the number of CG motifs. They're just 2 base motifs. And then look inside that locus for these, just a count, and then maybe patterns. So if they repeat, then I can get an account of that. And so just if I have like a protein on our list, I go find the genes associated with it, which is something that you can find pretty easily. It's not like an exhaustive search of all genes involved, but like the main ones that people have identified. Get those counts and then see if there are patterns there in terms of the number of CG motifs. I think like there's already some interesting results, but that's something I'm working on. I'll try to get that done this week.
Bradley, I think this section is going to be called the attempt to find the differentiation tree.
Well, yeah, I think that, yeah. So that's like the third part would be to map those findings to like the differentiation code or something like that. But that's actually something that can be kind of proposed. I mean, we have like I showed the 2016 paper where we have this worked out for cell size and cell or lineage tree. We don't have the single cell or genomics and proteomics, so we have to go with the whole organism and in genes, but we can actually propose maybe something about like these different proteins and things that change as we go. I don't know. I have to think about more about that. It might end up being kind of more speculative, but that's probably
That's
okay. Yeah.
That's okay. I didn't expect us to solve the problem.
Yeah.
Sure.
Well, I mean, yeah.
How much progress you guys have made.
Yeah.
There's an interesting switch that from in endothelial cells that goes from a non-motile cell to a moving cell, and it happens as soon as there's no pressure or flow on the endothelial cell, and it just switches cell type instantly. So there's a number of cells that do that, but that's the one I'm aware of.
Okay.
And it's I don't know. I have another picture of one in this PowerPoint that I've got of interesting cell things. I'll find it.
Okay.
Yeah.
Yeah.
The PowerPoint is 98 slides long, so it's a bit long.
Yeah.
So,
Yeah. I've had that problem before. So yeah, the other thing I wanted to talk about were the figure or the tables. So I'm working on putting together some tables. So like I said, the first table is table 1. This is where we have the different species, model organisms. We have the proteins here, and then we have, the counts for each. So this would be counts. And then table 2 might be like something like a linkage from the proteins to some of the genes, and then this count that I'm working on for the CPG islands. Table 3 would be something, I worked all this out yesterday in my head, and then I lost it, and then I put it down on a piece of paper, which I don't have with me right now. So that's great. Table 3 is in some combination of the data where I don't know what exactly we want to put in table 3, but there'll probably be 3 tables here I'm envisioning with some of the data. Maybe in this one we can have some breakdown of the annotations. So, like the annotations tell you something about the function. Like I said, it's imperfect, but we can actually maybe score them by core versus peripheral function, and then give a rationale for that. Why did we do that? And then show the differences there. So yeah, and then like maybe some figures as well. Maybe we put together a figure on what that looks like in terms of a differentiation code or a differentiation tree. What kind of clues can we draw? So if we had the ideal data set, which would be every cell in C. elegans with a genome sequence, a proteome analysis. We have some, annotations that are excellent. That's not what annotations are currently. Then we could say this is the way it would look. So we can actually, map that out a little bit.
Yeah, Bradley?
Yeah.
What drawing program are you using?
Oh,
Is it
well,
Bitsy?
no, I use something like Google Slides or PowerPoint to put together figures. And sometimes
what you were using a few minutes ago.
Oh, this is Jamboard. Yeah, so this is just like a, it's like what you use with you. There are a lot of different options for this, but I just use a Google Jamboard. I'm using a tablet on my end, and I'm drawing on it. So yeah, I have a tablet here. So that's that's what I'm thinking. And then of course we have the version 10 draft, which has a lot of the theory in it from Dick. It has some figures there. So we'll use that at the front and then put the analysis in, plug in a discussion with the figures on the speculation, and then that'll be it, I think. We also need to get some citations. So I have some citations. We'll put those in for the analysis and for some of the other
send
stuff.
it to me. I'll put them into an envelope.
Okay, yeah. So yeah, I think, and then Lukas said I could help you with gene counting as well. Yeah, we'll talk about that closer to when we get to that point. So, so for right now, Lukas, let's get all the different species down. let's get those together. I don't know if you want to put them in a table, just like for each. And
I'll
then
do the same thing. Like I'll put I'll make a table for the 4 model organisms, and then I say, like, let's say APC, KPC, all these proteins, and then I'll do the count for table 1, and then the protein. Maybe we could continue, and then I'll put I'll update the bioinformatics detail with V2, and then list all the proteins on my thoughts. So we could continue from that, and then we could use both of those data for the table 2 that you're working on, the gene counting thing.
Yeah. Let me suggest that you also rank them by count.
Yes.
Then do a plot of the count versus frequency of that count. If there's a break in that curve.
Sorry, I didn't understand the last part. Count versus frequency.
You bin them according to count. First rank them, then you bin them according to count. So you got how many in such a each range when you're binning. What was the count? And then we can see if that has any peaks or plateaus or anything like that.
Okay. Yeah, I'll do that as well.
Yeah.
Okay.
Yeah, I mean, I can help you with that. It's the matter of getting the right sort of version of it, or the sort of the visualization, which is hard to do because you have to kind of say, how do I want to present this? Like if it's like sometimes different ways of plotting it look better and they're more informative. So it's another thing.
Okay, so you understand what I was getting at?
Yeah.
Okay, great.
So this is another paper Susan posted on biomechanical, biophysical, and biochemical modulators of cytoskeletal remodeling, and this is for stem cell commitment or cell lineage commitment in stem cells. So again, this is where stem cells are going from a stem cell to something like a precursor cell to differentiated cell, and at each step, it's going through a cell division, it's remodeling itself. It's doing all these things. So a lot of these proteins are being produced, and then you end up with a new cell state, or in a new stable cell state, where it doesn't just move to another state. It stays in the trajectory of, say, neural precursor, neural stem cell, neural precursor to maybe a neuron.
Yeah, or it's epithelial to mesenchyme transition too. Like there's these switches, and they think they're very closely related genetically.
Yeah.
Just saying, that seems to be a hot topic.
Oh yeah. It's people want to figure out how that works. So yeah, we actually last week we talked about a paper in I think it was in Mammals, where they took a cell. This is the lineage commitment idea. They looked at different cells in a tissue, and they found that they had this heterogeneity of differentiation. So there were different. I guess they were either in different states or they were expressing different things. So even in the so-called regulative species, you have a lot of variation. So that's something to keep in mind, is that even if we had the perfect data set, we don't really know what this pattern will look like for each cell or each tissue.
You also have to be careful on the distinction between mosaic and regulatory. For instance, you take snails, they're highly mosaic during the embryonic stages, but as adults, they're regular.
Right. Just taking a note here. All right, yeah, so that's great. Well, thank you for the update. There are actually some papers that I was showing Lukas some papers on some of these things. So we have there are a lot of papers on phosphorylation. There's this one, protein kinases and phosphatases, and the Drosophila genome. So this kind of goes, this is like a classification of all the different protein kinases, and there are quite a few actually there are different families in the Drosophila proteome. So they have like a lot of, at least say 30 AGC kinases in this AGC family. So these are kinases that function in many intracellular signaling pathways, and they were classified based on their tendency to phosphorylate sites surrounded by basic amino acids. So they're making some sort of chemical modification to different things during transcription and during protein formation that are important for changing the function a lot of times. So you have all these different families, and this is kind of how they've broken it down. So they've actually made a comparison here in Table 1 between fly, which is Drosophila, worm, which is C. elegans, and then humans, which is the mammalian counterpart or the mammalian comparison. And so you can see that they have, for these different families, different numbers here. And so, we don't know what these numbers mean. They don't really trend in the same direction all the time, but they, it's a comparison. So we can say, well, there's a difference here in this species versus maybe this type of development versus just maybe random. So it's going to be kind of hard to say definitively what those comparisons mean. And that's kind of the point here. But I think it's important to build a table like this because you can see that, they're not all the same and that you have variation in some of these things. This is a paper on phosphorylation dynamics in human embryonic stem cells. This is the paper that Lukas referenced when we were talking about stem cells and the role of phosphorylation. So the reason I gave him all these papers is because, a lot of these proteins are involved in phosphorylation, and that function is distinct from, say, like the cytoskeleton, but it's also very important. So we know from this paper that in human embryonic stem cells, there's a lot of phosphorylation going on during early differentiation. So we know that some of these protein networks that exist in the cell, so you can actually do proteomics on the cell, and you can get these protein interaction networks, and those are published. We know what those look like. I wouldn't bring that data here directly to this problem, but it's important to remember that pluripotent stem cells do this thing called self-renewal. So anything that's totipotent or pluripotent self-renew. So they can divide infinitely and not die off. Whereas cells that are differentiated tend to have a limit where they have a certain number of divisions and then they go maybe towards apoptosis. So this is something where you get this self-renewal aspect. And then this is characteristic. You can find this in protein networks by looking at the different proteins that are interacting. And this allows, some of these networks also allow for remodeling during differentiation. So we have to go from the self-renewal phase where the cell is dividing and just self-renewing versus a phase where the cell goes through a division or goes through a cycle of RNA synthesis, and then actually changes its state or moves towards changing its state. So we don't really understand how this works in terms of all the mechanisms. People have looked at different mechanisms, which we've talked about. So they've been able to analyze the phosphoproteome of human embryonic stem cells, and basically they have some numbers. Usually the way they report this is that they'll take their whole number of proteins that they actually work with, identify. So they identified 5,222 proteins. 1,399 of those were phosphorylated, and 3,067 residues, which means that they were able to, these are out of the ones they identified, these are the ones that are phosphorylated. Approximately 50% of these phosphatases were regulated within 1 hour of differentiation induction. So this is a quick process, and so this is revealing a complex interplay of phosphorylation networks spanning different signaling pathways and kinase activities. So this is involved in a lot of different signaling pathways. The paper kind of gets into some of those. Among the phosphorylated proteins were associated with SOX2, which is something you see a lot in stem cell differentiation. There are other things, CDK1/2. This is something that controls self-renewal. So, a lot of these papers, they'll identify key genes or proteins that are involved in some process. And sometimes they'll also identify the network, but usually, it's based on genes because there's this tradition where you would say, okay, this is the thing that's responsible for this thing, maybe the protein. You go back, clone the gene, and do an experiment. Nowadays, of course, we have this systems approach where we can look at all these things in simultaneity. So, but people really haven't embraced, like, I'm going to replicate this network experimentally. I mean, I don't even think it's, in a lot of cases, it's really possible to do. But so that's that paper. And then there's the human phosphoproteome. So this is the functional landscape of what we see in humans. This is where we have all these, this phosphorylation across the human proteome. Phosphorylation is a key post-translational modification regulating protein function. So this is after the DNA has been turned into, it's been transcribed and translated and then transcribed. So those are two separate processes. During translation, you're just creating mRNA, and then after transcription, you're creating amino acids that then serve as the building block of the proteins. And so post-translational modification is where you're taking those chains of amino acids and you're modifying them. And this is what phosphorylation does. So it modifies the function of the protein. And so this is, they're involved in a lot of things, but, you have this specific role in differentiation. And so this is kind of gives a lot of information at this very broad level. So, I don't know if I don't think there's a paper on that specific mechanism with respect to, like, C. elegans differentiation, but that's kind of where we're headed, actually, in some ways, because we're talking about the proteins and seeing which ones are important. Final thing I want to talk about today is this paper. So Dick gave me an assignment last time, and it was about, if we have C. elegans and we have other species in Cantorhabditis, what is the relationship between their lineage trees? And I had said that there are differences in the lineage trees, which is true, but there's a paper that came out, I think, in 2019, so it's not that old, but this is Ralph Schnabel, who's done a lot of stuff in C. elegans and some other authors. And this is on comparing C. elegans and I think 3 other species. So this is called 20 million years of evolution. The embryogenesis of 4 Cantorhabditis species that are indistinguishable despite extensive genome divergence. So what they're looking at in this paper is I've identified 4 Cantorhabditis species. We have C. elegans, C. briggsii, C. romani, and C. brenneri. And these are just like different species of that Cantorhabditis clade, or that genus. And so they're related evolutionarily, but pretty closely. But they have different, differences in their lineage tree. They show more divergence at the genomic level than humans compared to mice. I guess that means that their genomic divergence is actually quite extensive within Cantorhabditis versus, like, between mouse and humans and mammals. So this
Bradley, are they interbreedable?
No, they're not. Yeah, they're so, like, in some Drosophila species, and they can breed. They actually can produce viable offspring. But I think C. elegans is pretty much C. elegans. There's no, I don't think there's any evidence of them interbreeding with other species of Cantorhabditis. However, the behavior and anatomy of the nematodes are very similar. So this is where you look at under a microscope; they look very similar. But this is something that we know that there's a lot of genomic divergence. So we present a detailed analysis of the embryonic development of these species using 4D microscopy analysis of embryos, including lineage analysis, terminal differentiation patterns, and bioinformatics quantifications of cell behavior. So further experiments support the notion that early development of all 4 species depends on identical induction patterns. Based on our results, the embryonic development of all 4 species is nearly identical. So this is where they basically have the same pattern of divisions and binary divisions and different tissue formations and things like that, suggesting that an apparently optimal program to construct the body plan of nematodes has been conserved for at least 20 million years. So these 4 species are related at the level of like 20 million years of evolution. So human and mouse are probably around that. It's, it depends on who is doing the phylogeny, but it's comparable to humans and mice in mammals. So this is a pretty deep for related species, in the same genus. It's pretty deep history. So this contrasts the levels of divergence between the genomes and the protein orthologs of the Canarhabditis species. So we have these protein orthologs that we can look at. So they're not all the same name. They're not all the same. Some of them are duplicates, and then they've diverged. So they're different. I have had to get back into sort of some of this like genomics and proteomics language. So they're orthologs and they're paralogs, and then they're homologs. And so those 3 terms are not the same, and they're just different ways of looking at these relationships. I can't remember what orthologs means, as opposed to like homologs and paralogs, but so that's yeah.
It's as if when you get a mutation, it's going to be generally inviable unless you get a second mutation which compensates for it, but then you get a different genome.
Oh, okay, yeah.
Right?
Yeah.
And it's sort of like building something. The director said, if something doesn't work, you take a piece out, you put a new piece in.
Yeah. Yeah. So yeah, there's so they go through a lot of these studies that kind of talk about the 4 species. There's some good figures in this paper. This is actually a good example here of what they look like. So this is C. elegans, I think at the 256 cell stage. So they're looking at the 12 cell stage here, where you have the cells, and these are like the 3-dimensional positions, and they're color coded by their fate, and because the this type of development you have in the nomenclature, you have like founder cells, and so those founder cells are sort of the basis for all the cells that descend from it. So you have like this green and this purple, and that purple cell is going to give rise to a bunch of cells that all have a fate that's either like in that tissue type or a series of tissue types that are related. So they look at the founder cells in this place here, and then 256 cell stage here. So you see a much denser cloud, and you see these arrows where they show kind of the different colors. Then this is the DIC microscopy on the left. So this is light microscopy that shows C. elegans, C. Briggsii, C. romani, and C. bruneri. So you can see they look similar. They have a similar sort of development where they come from founder cells and they proliferate, and then you have this. This is before the comma stage. So you're not seeing any formation of the body yet. You're seeing just the cells sort of massing. And, again, all these cells have a deterministic differentiation. In other words, they're going to divide and become a certain type of cell. And that's what they look like.
Bradley, it looks like if you could stop them at this 12 cell stage, and then mix and match between species to try to see if there's errors.
Yeah, you could do that, but more likely than not, they would just die. They wouldn't. Yeah.
That's delicate stuff.
Yeah, it's hard to say though. We don't know. Yeah, so these are the founder cells here. These are just versions of AB, A, AB, P, and then down there, and then the germline, of course.
Okay, can you show us the lineage trees?
I don't know if they have the lineage trees in the paper. Let's see. This is actually, these are distance maps that they use for the different, yeah, so they look at the distance map, which is the distance between cells. The A actually is what they call the OX distance map, which I don't know exactly what, this is only displays distances differences along the anterior posterior axis or the x-axis of the embryo. So we've done stuff like this where you take like 2 dimensions versus 3 dimensions and you look at their distance. And this is not the lineage tree, but this is like a distance map. So we do this, we've done this for different things. And then they just show this across species. So you get this, these differences in position, and you can actually characterize them that way. And then this C is, it reflects all 64 combinations of the founder cells, the 8 AB founder cells. So it's just the AB sublineage, and then their distance here. So they have this distance matrix. And this is kind of like, just kind of shows you that they're in different positions in the embryo. So this is the lineage tree. In this case, they're working from the AB sublineage, which is the anterior end, and they're just kind of looking at that. So I don't know if they have any other figures in here worth talking about. Yeah, I don't think they have any lineage trees in here mapped out.
Can you check if there's a follow-up paper?
I don't know. Yeah, we might. I don't know where to go for that right now, but
Web of Science.
Oh, yeah, I'll do it after the meeting, I guess. But yeah, it's 2019, so there might be a follow-up paper. Yeah, and so this is the last one where they talk about cell cycle length. Cell cycle is measured for the ABARPPPP lineage, which is pretty far down the lineage tree from the origin. This is where the cell is differentiating into the V6R hypodermal cell. So this is basically looking at cell cycle length and time. So the idea is that, it takes a certain amount of time for a cell to divide into its daughter cells. And so an ABA cell divides into an ABL and an ABAL and an ABAP or something like that. And it takes a certain amount of time. And so the idea is that between species, that time can shrink or expand. And so that has an effect on when that cell is produced. And if that's the timing is off, it can, maybe be like in a mutant where there's a mutation that affects the timing of that division. It can become non-functional, or it can do something else.
Then it'd be really worthwhile looking at the lineage trees, because it's possible that there are at least large parts of it that are what are called heterochronic.
Right. Yeah.
So it could be that the trees are similar, but they're stretched differently.
Yeah. Well, I know, mutants, sometimes you do get heterochrony, so not always like in a good way.
Okay.
Yeah. All right. Any other comments?
Lukas wanted to say something.
Yeah.
Oh, that was a misclick. I raised my hand by mistake. Sorry about that. Yeah.
That's okay.
Okay.
All right. So any other comments or questions before we go today?
I guess it's just nose to grindstone and ready for something.
Yeah.
Okay, and I'm sorry, I'm just out of it. I'm trying to do this candidacy project, and until I get it done, I'm like not even doing much around home.
Right. Well, good luck with that.
Thank you.
Yeah. All right. Well, if there's nothing else, talk to you next week.
Okay, thanks a lot.
Thanks.
Bye.
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 Have a good day.
Me too. Okay, now I'd like to talk about a few papers that have recently come out. It's on a topic called genotype to phenotype mappings. So there's a lot of literature in this area, and there's a lot of reading. If you're familiar with the work of Gunther Wagner, he's someone who's worked on this extensively. But this is a couple of papers where they talk about genotype to phenotype mappings more generally, and then they talk about it in the context of early life. So the first paper is on From Genotypes to Organisms, State-of-the-Art and Perspectives of a Cornerstone in Evolutionary Dynamics. And there are a whole host of authors here. Susana Manrubia et al., Lee Altenberg is on this, Santiago Elena, Pauline Hugaweg. I'm just looking at the big names. But there are a lot of people on this paper, and so they've thought about this quite extensively. This is in the journal Physics of Life Reviews, so this is sort of a physics of life take on it. The abstract reads, Understanding how genotypes map onto phenotypes, fitness, and eventually organisms is arguably the next major missing piece in a fully predictive theory of evolution. So these people are kind of taking the extended synthesis approach to evolutionary theory, but in general, the notion of fitness, some of the details are quite controversial in terms of how fitness is measured and how it's heritable and so forth. Also note that they talk about phenotypes and fitness and then organisms as separate things. So a phenotype is not an organism per se. A fitness is not an organism. They're all totally different things, and we're going to get into how those are related. So we generally refer to this as the problem of the genotype to phenotype map. So if we move into conceptual land for a minute, and we talked about this in the meeting, we have these networks of gene-gene interactions and protein-protein interactions. And we can collectively call this the genotype. So it's the parameter G. And in genetics, we'll often use a parameter G and then partition it statistically. So you'll have G by G interactions, and you'll have G by E interactions, which are gene-gene interactions and gene-environment interactions. So, but this whole concept of G, or the genotype, is quite broad. It could be any set of genes in the genome. It could be a specific set of genes, a specific circuit mapped to something else, mapped to some function. It could be the entirety of the genome, mapped to the entirety of the phenotype. And so then they talk about P, which is the phenotype. And so this is the phenotype. Again, it's a very broad network of things. It's not necessarily the organism, because the organism is going to be both G and P and some other things as well. So this is not the organism, it's just the phenotype. And one of the things, with phenotypes, we basically have to define them as maybe like a body, or it could be like a behavior even. But usually people think of, they think of the behavior as a phenotype, I guess. But again, you have the phenotype, you have the genotype, and the organism, which is the bridge between these. So if the phenotype is maybe like a morphological trait or a behavioral trait, those are both phenotypes where there's an underlying genotype. I'm not familiar with how people characterize phenotypes statistically, but what they're going to argue in this paper is that there's this mapping that occurs. And so there's a mapping between genes in a genotype, or proteins in a genotype, and the elements of a phenotype. So all these different, there's this old idea that one gene equaled one protein equaled one piece of the phenotype, but that's just simply not the case. We have these gene-gene interactions, which are this network down here in the G area, and then we have GE, which is where we have influences of environment, which we're not putting into this diagram right away, but it's basically that this network in the G partition, they're both interacting with each other, they're being expressed and they're co-expressed, and they're interacting at the level of proteins, and then there's this environmental interaction, which maybe turns genes on and off, or proteins get phosphorylated in different ways and so forth. Then there's a mapping to the phenotype, which means that maybe 6 genes interact and have different contributions of their variants towards the phenotype. That could be a behavioral trait or it could be a morphological trait. And then the organism is all of this put together. Okay, so that's what they're getting at. Now this bi-level network, as you see, between G and P, this is something we've seen in epigenetic landscapes or Waddington's landscapes. If we look at the pictures, I don't have a picture right now, but where you look at the underside of the landscape and you see all these lines coming out of little cubes, that's his conceptual genotype to phenotype mapping. So it's an old idea. It's been around quite a while. And like I said, this is something that is been worked on theoretically, but is, kind of hard to really put into practice. So this is where we're going with this. This genotype-phenotype map. We're still far from achieving a complete picture of these relationships. Our current understanding of simpler questions, such as the structure induced in the space of genotypes by sequences mapped to the molecular structures, has revealed important facts that deeply affect the dynamical description of evolutionary processes. So they're looking at evolutionary processes with something with dynamics. So they're almost looking at it as a dynamical system, but they don't think maybe they're not quite going there. Whatever. Empirical evidence supporting the fundamental relevance of features such as phenotypic bias is mounting as well. Yet the synthesis of conceptual and experimental progress leads to questioning current assumptions on the nature of evolutionary dynamics. Cancer progression models or synthetic biology approaches being notable examples. So there's this stuff that has been done on evolutionary dynamics and evolutionary game theory and things like that, where they've looked at specific systems like cancer progression or synthetic biology. This work delves in with a critical and constructive attitude into our current knowledge of how genotypes map onto molecular phenotypes and organismal functions, and discusses theoretical and empirical avenues to broaden and improve this comprehension. As a final goal, this community should aim at deriving an updated picture of evolutionary processes, soundly relying on the structural properties of genotype spaces as revealed by modern techniques of molecular and functional analysis. So it's interesting that they mention molecular phenotypes, because in the work of Gunter Wagner, which we won't talk about right now, he has this model of genotype to phenotype mapping, which is basically a DNA sequence mapping to an RNA structure. So RNA structures are basically a transcription of the phenotype or of the genotype to a phenotype that has some sort of secondary structure. So this is a loop. This is not based on anything in the real world necessarily, but this is basically your RNA molecule. This is your DNA molecule. They have these hairpin turns and other types of secondary structure. It's not the secondary structure of a protein, which is much more complex, but there are different secondary structures that exist. And so you map the DNA sequence to the RNA sequence, and then you have the secondary structure as well. So you have this genotype and you have this phenotype. And the RNA phenotype, of course, then that leads us to an analysis using a hypercube in terms of a neutral space. So we can look at the evolution of this RNA phenotype through the analysis of a neutral space. And I'll just use a cube for illustrative purposes here. But basically we have this cube, we have, I think we'll have 3 states here and 3 states here. So the idea is that you have to go from a state of 0, 0, 0 to 1, 1, 1. And you can take a number of paths to get there, and it requires a number of mutational steps. So this mapping will map mutations in DNA space to mutations in RNA space, and then you can have further mutations in the phenotype or in the genotype that transfer to the phenotype to lead you across this space. And so when we say neutral, we mean that there's no selective force, but of course selection can always act on this to guide it down the shortest path. In this case, every path is equivalent, so selection doesn't necessarily help you. So every path is like 3 steps, 3 mutational steps. And that's, something that if you had a more complex space here with more, with clearly shorter paths and clearly longer paths, then selection might help you get there faster. But in this case, it's a neutral space because every possible path is the same length, same number of mutational steps. And so you can see that this allows us to think about the phenotype very concretely, the mapping from genotype to phenotype, and then the evolution of that phenotype in mutational steps, which can be quantified. And then we can think about things like neutral evolution or selection or whatever. So this is the abstract of this paper. This basically talks about, this relationship between genotype and phenotype. And of course, if you have an RNA molecule as your phenotype, that's great, that's convenient. But a lot of real-world phenotypes are a lot more complicated than that. They're complex, they're not discrete, they have a lot of discontinuities. There are a lot of things in between, say, like a DNA sequence and like an embryo, a mammalian embryo, say. There's a lot of stuff in between there that we need to put into that model. So we might have, say, for example, a genotype to proteome type to structure type to phenotype map or something like that. We could have these models could be several levels deep. So and this is something I've been thinking about with respect to some of the things we were talking about in the meeting. So we have this genotype here, which of course is just a kind of a nondescript lake with a network in it. Right? We don't really know what this network looks like. It's just kind of like interactions. We have our gene, and gene environment interactions here. We have the environmental effect here. Then we might have, well, we don't have that arrow. We have a proteomic or maybe an RNA phenotype lake. This is, I don't know what you'd call this, RNA type or something. And there you have secondary structure and you have sequence structure. So you have 2 different forms of complexity. Then you go to your proteins, or, that has like a lot of interactions, protein-protein interactions. And then finally go up to the top level, which is the phenotype, which, you have all the sort of the interactions that occur at these different levels. You have transfers upwards, and then those will be sort of summarized in the phenotype. And again, this is going to be a hard sell because we don't know what the contributions are. We don't know what maps to what. Phenotypes sometimes are very easy to sort of discern, and sometimes they're not. So it's a very hard thing. But anyways, this is what it's, this is the sort of the logic of this approach. So they kind of talk about GP maps as if they're easy sometimes, but in a broader ecological context, the way in which generic properties of the GP map shape adaptation have been really explored. So this interaction between the environment and the genotype, or the environment and it could be the RNAome or the proteome or even the environment and phenotype. There have been a lot of arguments in evolution, for example, the level of selection. Is it at genes, or is it at the phenotype, or where is it? For the sake of argument, I'm just going to say it's at the level of genes. I think that's a good way to sort of characterize it, because the phenotype, it's extremely hard to know, what the effects of selection look like when you're selecting on a phenotypic trait. So if you were to domesticate dogs and you were to breed for specific traits, you're not breeding on the phenotypic traits per se. The phenotypic traits are the outcome. You're actually manipulating the genotype. So this is where, you kind of get into this issue of, like artificial selection and what are the outcomes of artificial selection. So it's a very interesting set of issues. So this paper is really long, and I'm not going to get into the whole paper. This review has a number of parts. There's the introduction. There's section 2, which puts into perspective how relevant the generation of variation is in the evolutionary process, and introduces important biases arising from the inherent structure of genotype spaces. So this is where the genotype spaces introduce bias into, say, development, or into the phenotype. How genes are expressed can affect what the phenotype looks like. So in developmental, in Devo, one of the main arguments has been like if you have a promoter that enables expression of a set of genes in different ways, or a set of promoters, that if they work one way versus another in two different species, you end up with two different modes of development, or you end up with maybe a trait that has a longer growth period, or a shorter growth period, those sorts of things. And so, we have these biases that occur both in development, in phenotype, but also their sort of origins are in the genotype. The second part comprises sections 3 to 6, where we discuss conceptual approaches to the static properties of GP maps and their dynamical consequences. So these GP maps are both static and dynamic. If we look at like this, it's pretty much a static representation. I'm just showing you the levels, but they're also dynamic systems in the sense that they evolve, and that they evolve. Sometimes they evolve independently. Sometimes they evolve in the same direction. With genotypes, and if the phenotype is something like a trait, a lot of times a genotype is a very, there's a very tight mapping between genotype and phenotype. But if it's something like a behavior, or like culture, or cultural evolution, then it can actually decouple quite a bit. And so this is something to think about when you think about the phenotype. What is the phenotype, and how are we characterizing it? So, they talk about the GP maps as static and dynamic entities, as well as the evolution of GP maps themselves. So these maps can evolve as the connections between the networks underlying them evolve. The field is broadened. Several aspects have been addressed in previous reviews, so we only briefly summarize topics dealt elsewhere. So this is just a review. We will succinctly present computational GP maps. So this has been done in computer science and A-Life and in genetic algorithms quite a bit. They work from sort of a genomic representation of a problem. So they'll take a problem and they'll encode it as a set of binary strings, and they'll treat that as a genotype. And that genotype is expressed, so certain bits are turned on, certain bits are turned off, and then that results in a phenotype. And so you can have these computational maps that lead to certain phenotypic outcomes. And then this is, of course, can lead to topological properties of the space of genotypes. So you can actually look at a genotype space, all the different states. So it looks a lot like this cube, which I showed in slide 7 here. You have this sort of cube that arises from the genotype being expressed in different ways. And there are these mutational steps that can be taken to transform from one phenotype to another. So there are a lot of ways you can do this. Computational methods, I think, are particularly useful here because the empirical space is so fuzzy, and we're dealing with such a large area. So I would definitely start with some sort of simple computational model and then move from there. They give a mean field description that incorporates essentials of GP map topology that clarify major dynamical features. Then it finishes with a derivation of equilibrium properties in the context of statistical mechanics and some applied examples. And then section 6 discusses the evolution of GP maps themselves by means of 2 examples. One is a scenario where a multifunctional quasi-species emerges, which is something that you see this is like evolutionary dynamics jargon, and I can't quite remember what a quasi-species is, but I think it's like a class of something. And then a model of virtual cells incorporating the evolution of genome size. So this is another model of the phenotype from a genotype. And then in 7 and 8, they talk about empirical GP maps. So this is where you have real biological systems and what those look like. And so this includes a formal discussion on how phenotypes can be inferred from genotypic data and fitness assays, and a discussion of the intimate relationship between fitness landscapes and GP maps. So this paper goes over covers a lot of ground, reviews a lot of ground. The fourth and last part presents a mostly self-contained overview of open questions. And so the field faces a number of difficulties, as we've just discussed. So this is something that, if you're interested in this area, it's a good paper to read through. Even if you're not, I think it's good to think about some of these problems. And Stephen Jay Gould, of course, thought about these problems. So he was very critical of the viewpoint that a large part of the variation in organisms is isotropic. In other words, it's not biased in one direction or another. So Stephen Jay Gould was critical of this viewpoint and expressed it as follows: variation becomes raw material only, and as an isotrophic sphere for potential about the modal form of a species, only natural selection can manufacture a substantial directional change. So this is something like we're talking about with the bias in these models. So you could just have a genotype to phenotype model where one gene mapped to one piece of the phenotype. You could do this in an additive way, but that's not actually what biology gives us. Biology gives us variation to act upon, and then to form these biases that allow us to express things like neutral processes or natural selection. So this is something that, it's not necessarily directed evolution in the Lamarckian sense, but it's this sort of bias that results from these different levels being pushed in different directions. Sort of almost like you can think of it as path dependence, maybe rather than directional evolution. But the outcome looks like something maybe like directional evolution. So there are different models that people have introduced. Maynard Smith introduced the notion of mapping from a genetic space to a molecular structure. I think we talked about that a couple of weeks ago in a classic paper. And the idea is a network that links viable genotypes. This is a resolution of an evolutionary paradox pointed out by Salisbury, where the number of possible amino acid sequences exceeds by many orders of magnitude the number of proteins that has ever existed on Earth since the origin of life. So you need to have a subset of those potential amino acid sequences. The information is in restricting that to a certain alphabet or grammar. And so how does that happen in evolution? And so this is kind of the attempt to make this non-Lamarckian. And so this is where this comes from, part of it as well. They talk about neutral networks, which is this ensemble of connected genotypes with the same fitness. And as we saw with the RNA, could be an RNA phenotype, including those with identical phenotypes. So you can actually set up your neutral network as a series of genotypes that map to phenotypes that look the same. So they're these phenocopies of each other, but they have different underlying genotypes. There are a lot of ways you can do these neutral networks. The point being is that you have the space where you can, have these neutral processes play out. So here are a couple of model systems here. This is an RNA molecule secondary structure. These are some of these hairpin turns in a model where they give the sequence, but they also give the structure. So the sequence is derived from the genotype, but the secondary structure is also a feature of the phenotype. The HP compact, which is this protein sequence here, and it has nodes that are connected. Toy life, which is this toy polymerase, toy gene, and then expression of the toy protein. Then we have these simple effective GP models, which is where you just have like these strings that are expressed at different points. You have stop codons at different points that truncate the sequence in different places, or you have this with actual nucleotide bases, so you can actually edit them in different ways. A polyomino, it's like a domino, but So this is where you have these different geometric configurations with different sides, faces, different values, and you connect them together in a way that's valid, and that kind of restricts that possibility space down. And then you can examine what kinds of sequences you get from that. So not every configuration is allowed in the game, so you have to fix, I guess you have to interact sequential numbers, so 1 fits to 2, 2 fits, or well, I guess 2 fits to 3, we don't have that. We have a 3-face fitting to a 4-face, and a 2-face fitting to a 1-face, so there's a transition here. You can't combine non-likes, so you can't combine a 0 and a 0. So you end up with a subset of these shapes, and you can characterize the sequence here as the genotype, and this is the phenotype. And then biomorphs, which are these models G. This is Richard Dawkins' biomorphs, where it's a genotype with a few parameters that define the generative rules of the structure. So you take this genotype on the right, you can generate these phenotypes on the left. So then we have genotype networks. This is generated by assigning viable genotypes at random with a certain probability to binary sequences of length L equals 8, so a certain length, a certain probability. This shows a network of different genotypes. So this actually shows like lethal genotypes here, viable genotypes in green, and then recombination centers in blue. So you can connect genotypes together in a network. This shows kind of how this happens in different simulations at different mutation rates and produces these genotypic networks. And so that leads us to box 5.3, genotype spaces as a network of networks. So this is where we have networks that then form networks of their own so that they're connected. This is something that we see there's an area of network science where they kind of do this sort of thing, like they're meta networks or something. I can't remember the name right now, but one important motivation for these type of networks has been the finding that robustness, synchronization, or cooperation lead to different behaviors when studied and isolated or in interconnected networks. So if you connect things together, if you connect networks together into their own networks, you can actually look at things like synchronization and cooperation. Whereas if you just studied interconnected networks, one versus the other, you wouldn't be able to understand those things. The main reason for the change of perspective has been to realize that many natural systems, beyond displaying a network-like organization, are also made of interacting and competing networks at very different scales. So we have these different networks in our organism. We have molecular networks. We have, protein interactions, gene-gene interactions. We have molecular interactions, and when we're building, say, like a tissue or some sort of phenotype, we have organismal networks. So all those networks are embedded in an organism, which is then embedded in a network. So to get at all these levels of network, we need to have the same type of model that we have for our phenotype to genotype mapping. So we have these sort of layers, and it's vertically organized. And this is one of the reasons why we do this, because it allows us to get at some of these interactions. So this is a multi-level network, as opposed to just like singular networks. When I said meta networks before, I meant multi-level networks. But anyways, so the extent to which network science can foster our knowledge and comprehension of the evolution and adaptation of heterogeneous populations in an ever-changing biosphere is a relevant open question. In particular, the theory of competing networks can be used to analyze the evolutionary dynamics of populations in a space of genotypes that can be regarded as a network of networks. So in this viewpoint, population evolution is described as a competition of resources for resources of a certain kind, where the competitors are whole networks instead of independent nodes. So again, if we think about like a model of population genetics where we have a single node, an organism being that single node, or a genotype being that single node, we can actually think of that node then as this network within that node. So the network changes if there's mutation in that network, or if there's mutation in that node, we can represent it more clearly. We can really understand kind of what's going on inside the node. and then, of course, understand how competition unfolds. So this is kind of what they're doing here. So finally, I'd like to talk about virtual cells and evolutionary dynamics. In the RNA world model described herein, molecular phenotypes were determined for pairs of complementary sequences, positive and negative strands based on specific structure motifs. Under evolution, the following phenotypes emerge: self-replicases, parasites, helpers, stallers, and junk. So these are different classes of phenotypes. This is something you kind of find in cellular automata in the game of life, where you see gliders and other signs of structures. So this is not something that, this is very compatible with that approach. But they're also building these virtual cells. And so they're trying to get a sense, I think, of the virtual cell and what it looks like. This is at the origin of life. So you're trying to build different classes of things that do different, that will fit into a sort of a functional network within the cell. So here you have a diagram. So this is virtual cell and evolutionary dynamics. Around here is the scheme of a virtual cell. So you have some sort of ion pump. It's pumping things into the cell. You have these different reactants in the middle of the cell. You have the end product, and then you have transcription factors that bind to genes and express them. And so this is a whole sort of metabolic cycle within the cell. This shows the genome size in gray. So this is the gray function. Underneath the function is the gray. And then this red function, which is showing common ancestor through time. Red line is the average fitness in 3 standard environments. So this is the fitness versus the genome size. Shaded area, gray area predicts genome size, showing initial genome inflation, which is here, followed by a streamlining or gene loss. So maybe what's happening there is that you have a lot of genes, and they're encoding for a lot of things in the virtual cell, a lot of different components, and then those maybe get reduced as it becomes more efficient. So there are things that are maybe shared genes, and they're expressed in different ways. That's just my interpretation of the graph. I'm not really sure what's going on there, but we can see that there's this comparison between fitness and genome size. So fitness is actually fairly low for a large genome, but it increases as the genome shrinks down in size and finds sort of a stable equilibrium. Mutational neighborhood of the ancestor in various time periods, which is C, color-coded according to inset. This is the inset of this here, and we can compare the two with the fraction of mutants versus the residual fitness fraction. This leads us from an initial fitness distribution, the shaded gray area, to a more pronounced U shape, which is this fraction of mutants under this residual fitness fraction. So it kind of goes down and then up, going from 0 to 1 as a residual fitness fraction. So I guess the residual fitness fraction is neutral mutations are 1, deleterious mutations are 0, if I'm understanding this graph correctly. In any case, you can see some of the evolutionary dynamics here. Now on to the paper on early life. So this is Christoph Adami and Nitash C.G. And they're from Michigan State. And they do a lot of stuff with, Christoph does a lot of stuff with biophysics and A-Life, especially like the Avita platform. He was one of the people to develop that. So this kind of talks about this original paper that we talked about previously. So in the target article, the authors share a broad overview of computational and theoretical investigations into the nature and properties of complex fitness landscapes. These fitness landscapes are, as the authors emphasize throughout, complex maps from a genotype space to a phenotype space. This map is necessarily complex because, for the most part, there are many more distinct genotypes, in a sense exponentially more than there are distinct phenotypes. So there are a lot of genotypes that exist that might be inviolable as phenotypes, or you have these phenocopies where the phenotype is essentially the same, but there are different genotypes underlying them. So you have like different types of neutral mutations that don't contribute to the phenotype. You do have deleterious mutations, and you do have beneficial mutations, but those beneficial mutations aren't always expressed in the given environment. So, I mean, there are a lot of ways in which phenotypes can be similar, but genotypes can diverge. So this makes this map even harder to really kind of understand. One consequence of this feature of the genotype-phenotype map is that the fraction of functional sequences, those that have a particular phenotype, is very small compared to the number of possible phenotypes. In fact, this density of functional sequences can be used to quantify the information content of the sequences. It's pointed out by Shostak. He defined the functional information. A molecule has about a particular phenotype E in terms of the fraction of sequences Fe that carry the phenotype at level or higher as this. So this is the information, and then this is log F. So this is basically an information metric on this. So it's a fraction of sequences that carry the phenotype, and there's a threshold for the phenotype being carried. So it has to be like, the phenotype might be cryptic, or it might be very distinct, and it has to be at a certain level. And then so this just shows this information measure that they're using for this. It's basically negative log. So that tells us something about the information. And then if the sequences are written in an alphabet size D, so that means like the bases that you have. So for proteins, you have 20 bases, right? You have 20 amino acid types that can be combined. Or in DNA, you have D equals 4, you have 4 nucleotides. So this is how this is calculated, then limited to sequence length L, then N equals D to the power of L, and then that's our information measure. So we're just multiplying L into the information. N actually is, so N equals D to the power of L, so you have log D and then L minus that, and then that's your information. So then log N is just the microcanonical or coarse-grained approximation of the Shannon entropy. So we've seen this in Shannon entropy, basically. It's a similar thing. An approximation of Shannon information content of a biomolecular sequence. So a lot of times people will calculate the Shannon entropy of some sequence and then use that to characterize the genotype and its information content. But Shostak's measure is a convenient way to quantify how much information about performing task E is stored within a sequence. So this gives us a good measure of this genotype to phenotype mapping. And so then they're applying this to Avita, and this is where you're building these self-replicating programs that have a certain instruction length. The instruction length can be reduced to this symbolic diversity that you're going to measure with this information measure. So using the number for n and 2, which is this equation up here, and then using the phenotype colony forming as a threshold, so it has to have a colony forming attribute, and whatever that looks like. I guess you could, in a computer program, define a colony forming phenotype very precisely. It reveals an information content of 5.91 mers. So it's like a mer is just like the number of characters. So basically, it's 5.91 characters for this information that has the information content of the phenotype. So basically, in Avita, this is it's a very easy way to do this. You have an instruction set that produces sort of these behaviors or these competencies. You then have this, they can be reduced to a sequence, then that maps to some behavior in the program. And then here we have this colony forming phenotype that we can evolve. And then once we've evolved it, we can characterize a genotype in terms of what instruction set is needed, or the minimal instruction set, I guess, is needed to produce this behavior. And so this is important in early life because we get these sorts of behaviors that emerge very early on that we want to know, like, what was the minimal genotype necessary for this type of phenotype. So this shows a network of this largest cluster containing over 94% of the replicators for the L equals 9 landscape, with edges drawn between 1 mutant neighbors. This graph consists of 5 sparsely connected groups of tightly connected clusters, as you can see here. And so then so this is different calculation. So the information content of the replicators is 5.77. We're using the same equation. Replicators differ vastly in their robustness, so replicators are sort of the vehicles for replication. So they differ vastly as measured by the number of one mutant neighbors that are also replicators. Well, the most robust replicator is 74 viable one mutant neighbors, so they're using this network analysis to understand like what the effective replicators are, because they're self-replicating. You can measure these sorts of lineages. We can test how information is encoded in replicator I by analyzing the density of replicators and mutational steps away from that sequence. So this gives us this equation here. And then in equation 5, there's a number of viable replicators at mutation depth k away from the target sequence I. So we go back to this cube, or this hypercube analog that I mentioned before. You're basically trying to figure out what's so many steps away from sort of the target, or like from an initial sequence. So you're kind of building this kind of network where you have sort of this starting initial state and then sort of an end state. So this expression is the number of viable replicators at mutation depth k away from the target sequence I. Okay, so then there's the number of this denominator here is the number of possible mutants up to distance n. So that function PIN is the density of replicators at distance up to n from sequence I. So you're counting up the density of replicators across the diversity of sequences. Plotting this information against n reveals that information is encoded within the sequence. So these are the graphs here. You can see that this is the fraction of functional sequences on the y-axis, the mutational depth on the x-axis, and you can say as mutational depth increases, the fraction of functional sequences was down to -6, but I think that's the log. So yeah, so it's going in this downward slope. The red line is the theoretical limit, so nothing should be below that. And then these black lines are the different examples that they got from the program. The red line indicates the density decay if sites do not interact, no epistasis. So epistasis are these gene-gene interactions, and most of them are exhibiting epistasis, so you see them above the red line. Okay. Okay, so that's all for that paper. It looks like that's was a short set of experiments, but I think you can see the relevance to this relevance of this approach to different problems. We didn't get to the open problems. So finally, we go back to the first paper. We can talk about some of these open problems. And there are a lot of them listed. I'm not going to go through all of them in the interest of time, but I'm going to talk about a few. So one of them is 9.1. So if we understand this GP-map architecture, we might ask the question, is it universal? So we might ask the question, is it always a matter of mapping from genotype to phenotype? And we saw in the mathematical formulations that there is a information content of the genotype that needs to be carried over to the phenotype to make it viable. We also know that certain combinations are we were biased towards certain combinations of genotype to produce a phenotype, and only those certain combinations that are favored maybe by viability criterion or by selection or by neutral processes or by development are going to be viable. But is it universal? Does this always need to happen? And so they talk about this to different biological relevant situations. Models such as toy life and virtual cells should be further studied if we want to explore issues relevant to synthetic biology and some other systems where we can really manipulate things and get a good account of this. So another area is constructional selection. And so they point out here that genes not only provide material for the generation of the phenotype, but also provide degrees of freedom for varying the phenotype. A gene duplication, or de novo gene origin, thus differs from a point mutation in that it increases the degrees of freedom of the GP map. So different types of mutation matter, and gene duplications can increase the number of degrees of freedom of the genotype and thus the phenotype and that whole entire mapping, and thus adds new variational properties of the genome. So this is something that we see a lot in eukaryotic reproduction, where we get gene duplications and deletions. Any variational properties of a gene that is associated with the gene being retained in the genome can thus become enriched in the GP map. So this is where, there's this, if genes are retained, it can become a larger component of the GP mapping. It becomes more important to the phenotype. And so this is something that, is an interesting area of research. I think we can do it with simulations, but also with very simple model organisms as well. And so differential gene duplicability, which is where some genes are replicated, others are not replicated. This is something that people have investigated and what its consequences are for organismal evolvability. And we can build quantitative models to look at these variational properties. And in evolutionary computation, they call this constructional selection, but you can look at it in other ways as well. And so finally, there are these entropic evolutionary forces. And so we talked a little bit about information content and entropy in the other paper, but there are these different entropic phenomena. And so this results from evolution along neutral networks. So a couple of these are subfunctionalization, where you get different functional modules, constructive neutral evolution, which is something we talked about. Erland Stoltzfus has introduced this general concept of entropic processes that produce greater genetic complexity to treat simply because there happen to be more complex ways to generate a trait than there are simple ways. So this is where you get this, you start off with this simple way to generate a trait, and then as you move along in evolution, ways become more complex, and this is the sort of entropy. You also see a focus on non-optimal phenotypes. So this is where you get a lot of, when you have weak mutation in a population, this is an exact analogy to statistical mechanics, which is something that some people call free fitness, which is the sum of the fitness phenotypes and the sequence entropy. The analog of temperature is the inverse of population size. They use that type of statistical mech model to kind of understand what's going on in these populations. So for small populations, evolution gives rise to non-optimal phenotypes that balance fitness and entropy. There's also developmental systems drift, which is where primary sequences may diverge between species, even while the same developmental outcomes are maintained. And so it is predicted for small populations that the effect of sequence entropy is that populations develop isolation more quickly. So those are some questions or some issues that are raised by the first paper that relate to the second paper. And this is an interesting area. I think it's very useful for a broad range of things, and hopefully we can follow up on this soon.